So the topic for today's webinar is Mirrors to Solve Stress by Dr. Neeti Sanan-Mishra, Group Leader, International Center for Genetic Engineering and Biotechnology, New Delhi. Dr. Neeti Sanan-Mishra graduated from Delhi University with a bachelor's degree in Botany honors. She completed her M.Sc in Life Sciences from Jawaharlal Nehru University and went on to pursue her Ph.D. in the area of Molecular Plant Physiology from JNU. Thereafter, in 1998, she became research scientist at Center of Plant Molecular Biology, University of Delhi South Campus. She joined ICGEB in the year 2000 and currently she is leading the Plant RNAi Biology Group. Her group's research interest is towards understanding the miRNA networks involved in regulating the plant development in response to virus infection, high temperature, and salt stress. The group is using bioinformatics along with the traditional molecular biology and biochemical approaches to understand the miRNA regulations operative during development and stress. She has several awards and patents to her credit. She is also on the editorial board of scientific journals. Dr. Neeti has published several research articles in the reputed international peer-reviewed journals such as PNAS, Scientific Reports, Frontiers in Plant Science, Plant Molecular Biology, and Virology. We are highly honored to have her with us today. I now request Dr. Mishra to please share her screen and begin her talk.
Good morning all, and for those who are in India, and thank you very much for having me on this seminar series. So is my screen visible now?
Yes.
Yes?
Yes.
Okay. So thank you all again for joining me to listen me on this topic, which is MirROR, that is microRNA regulation of response to salt stress in plants. And at the outset, I must thank the team Bioingene for organizing this talk, and all of you to join in to listen to my views on what a response of plants to salt stress is. And this is more so in the time of today's pandemic, when there's a lot of social distancing and we are all far from each other, and viruses have clouded our mind. Now, other than virus, the most important thing that has crossed everybody's mind is food, because as soon as the pandemic was officially acknowledged and a lockdown was initiated, people started thinking about food, about resources to keep themselves fed during this time. And also many of us were thinking about those who don't have the resources and how to feed those people. So underlined here is the requirement for food. And one of the steps to mitigate this hunger is by growing more food. And this is where the challenge lies in front of the scientists today, because when we talk about food, we have to produce it in a present environment where there is a lot of climate change, there is an expanding world population, and there is a lot of environmental degradation. So all this is producing a lot of stress on the plants, on the agriculture and the produce. Also, it is highlighting the importance of food for human welfare, because directly or indirectly it affects the human health. And when we talk about crop plants, within the crop plants, rice happens to be the most used cereal. It is not only a cereal, it has become a part of our life, part of our rituals ever since it has been domesticated. It is no longer considered just as a food. It is very much a commodity, and is culturally a very important part of human existence. But what is the future of rice now? Oh, I have a screen which is missing. So what we need to understand is that rice is mainly grown in these Southeast Asian countries, and these are the ones which are facing a lot of climate change. And as a plant, paddy or the rice plant is considered most sensitive to stall stress during the early stages and the reproductive stages. And this directly impacts the grain yield production of rice. Now, the global climate change is produces stress to the plants. So what do we mean by stress? Stress is when the ambient temperature or the ambient requirement for a plant to produce, to grow in a healthy atmosphere and produce seeds is disrupted. And this can happen if we have one of the environmental factors, which is either too much, comes in too soon or too late, the timing is changed, or is too less. So example, in case of water, which leads to drought. And all of these impact the growth and the productivity of the plant, which impacts is directly seen in case of the diminished quality and the reduced yield. Specifically, if we look at the higher temperatures, it has been seen that for every 1 degree rise in temperature, the yield is supposed to decline by about, is predicted to decline by about 7%. And the impact is even more when we talk about the nighttime temperatures. Well, this is another line of study, and probably I will mention this during one of the slides later in the talk. So, how does this impact rice? Because if we look at a country like India, then we can see that the rice plant is growing almost everywhere, right from the northern tip to the southern tip of the country. And there is rice which can be cultivated as an irrigated dry upland rice. There is low rain fed lowland rice, which is mainly rain fed. Those wet rice which is growing in the flooded regions, and there's also deep water rice which has the capacity to keep the heads above the rising water table. So the rice plant can grow in a variety of regimes. Yet I say that whenever there is an environmental fluctuation, it will impact the productivity of rice. So what we need to understand is that there is genetic information in the genome or organization of rice plant for making it able to adapt to grow in a particular niche, whether it is from overwatered to underwatered regions. The capacity is there, but depending upon the niche or depending upon the area where it is growing, it needs the plants have adapted themselves, and hence whenever there is a fluctuation, an impact is perceived. Yet there is ample potential in the plant to be able to grow in one of these changing environmental regions. And to understand this, lot of efforts have been done by research groups all over the world. And if we look at the genome sequencing of rice, which started somewhere in the early 2000s and has progressed since then. And now we have more than 3,000, 4,000 genomes which have been sequenced, and a lot of information, different varieties are there. But why I am showing this timeline is that we need to understand that while this research had started in 2000 about sequencing the rice genomes, understanding or reading the genome, identifying what is written there, parallelly around the same time, in the 90s, began the revolutionary discovery of RNAi or RNA silencing. And 2002 was the year which has been acknowledged as the year of RNAi by science. So the two things ran almost in parallel with each other. And now, after almost 2 decades, we are at a position where we understand that the classical gene expression, along with that, there is a silencing mechanism that is happening. So the information which is written in the genome is transcribed into the RNA and translated into the protein. But some of these RNAs have regulatory function, and then they can go and they act as switches to switch on or rather switch off gene expression. And this forms a important part of silencing. And it is it is not expression alone or silencing alone which is important, but a balance between the two, which will govern or which will fine tune the genetic machinery or the genetic program of a plant or of an organism, so that it can grow and thrive properly in its natural environment. And this area is still a black box. A lot of information has been obtained, and we are still on a path of discovery to understand what is silencing and how a balance or a dynamic interaction between this expression and silencing machinery is maintained inside the tiny cell, so that the optimum responses can be maintained. Now, if we look at this RNAs or the regulatory RNAs, they are a big family. Within this comes these non-coding RNAs, of which there have been many classes which have been recognized. And within this class lies the 20 to 25 nucleotide small regulatory RNAs. And one of the major class of these small regulatory RNAs are the microRNAs. And these microRNAs are the ones which are responsible for regulating the developmental phases of an organism. And if I talk in the terminology of plants, making us understand why a rice plant will look very different and grow very differently than, for example, another cereal, say maize or wheat plant. Now, for those who are new to silencing, I will just very briefly cover the basics of what microRNAs are. Like I said, they are endogenous. That means they are coded by the genes. So there are specific genes present in the chromosomes which will code for these microRNAs. They are transcribed as larger transcripts, which are these single-stranded molecules are heavily or compactly folded and are called the primary microRNAs. From one of the folds of the primary microRNAs, a hairpin-like structure is processed out by the action of specific proteins, mainly the main slicer being the dicer-like protein or the DCL. And these hairpin structures are known as the pre-microRNAs. These pre-microRNAs are further processed to remove the duplex, the hairpin loop here, to form a duplex structure. And this microRNA star duplex is then methylated, and then all this process in case of plants occurs within the nucleus of the cell, and the mature microRNA is transported out into the cytoplasm, where it associates further with another complex of proteins to bring about the function, which in case of plants could either be cleavage or a cutting of the target mRNA so that proteins cannot be formed, or these are translationally repressed so that the translation machinery is blocked at one of the processes and the proteins cannot be formed. Now, a lot of research and lot of exciting results have happened, and we know the other molecules, the helper proteins, the partners which are involved in this whole process. This is a highly regulated process. It may be sequence independent, but then there is a battery of proteins which are tightly regulated, and they will regulate how and when and why will a particular transcript be processed as a microRNA. And once it is released into the cytoplasm as a functional molecule, on what transcripts it will act and how it will bring about the function. Now, this later or the functional part, of course, is a sequence dependent process, whereas the processing happens to be a sequence independent process. So overall, or generally, it is recognized that microRNAs repress the expression of target genes by either cleavage or translational repression, which form a part of the post-transcriptional gene silencing or the PTGS pathway. And there is another component which will go and switch off the transcription at the genetic level and will form a part of the TGS or the transcriptional gene silencing pathway. Okay, Now, just for people who are from the non-plant background, microRNAs are also found in animals, in insects, and all other eukaryotes. But there are similarities and differences in the pathway. And as a textbook knowledge or a basic differences between the two is, whereas the complete processing from the mature from the prior transcripts to the mature molecule occurs in the nucleus in the plant cells. In case of animals, it is the precursor molecule which is exported into the cytoplasm, is processed here, and then it gets associated with the functional protein complex to bring about mainly translation repression. MicroRNA mediated slicing is not known in case of animals or insect cells as of now, whereas both the mechanisms are present in plant cells. So this is just a very preliminary, a very basic textbook kind of knowledge to differentiate between the kind of the processing and the functional differences between the microRNAs. The molecules, however, remain the same. They are endogenous, and they can act on the endogenous transcripts. So when I say that microRNAs are produced from genes, we all understand that genes, there can be multiple copies, there are alleles, so on and so forth. And the same exists in case of microRNAs. There can be multiple copies of this of the MIR gene spread across different chromosomes, and hence different precursor molecules can give rise to many microRNA molecules. Now, there could be inherent or differences between these precursors at specific positions, but there are certain positions specifically from position 1 to 10 which remain conserved, and there could be differences further downstream, it being a 24 nucleotide molecule. And all such sequences which have similar structures are normally grouped together and are considered as members of a particular family. Now, this is important as I will come to it later, because normally the transcripts belong to large family members, and it is presumed that during evolution, the microRNAs have also evolved in such a way so that there are multiple members of the microRNA families to regulate multiple members of the transcript families, and we are still in the process of learning and understanding how this regulation and this coordination is brought about. We have some knowledge, but there is still a large amount of gray area which needs to be, the mist needs to be removed. Okay, so coming to how do microRNAs act, like I introduced that they can act through the PTGS or through the transcriptional gene silencing or the TGS pathway. The PTGS pathway will include mRNA destruction by cleavage, and the other would be the repression, which will can occur through several means. And the transcriptional gene silencing is mainly by regulating the chromatin structure through methylation. And without getting into the details of the two processes, this is just to give a brief idea on how the different microRNAs act. So now, knowing that microRNAs are there and they can repress transcripts, the question becomes, what do they regulate and why do they regulate? Now, if we look at plants, the entire morphology of the above ground or the underground parts, which can be quite variable depending upon the plant species under study or under consideration. MicroRNAs can regulate almost every aspect of plant growth, whether it is development, we talk about right from seed formation, cell division, organ identity, polarity, the vascular development. And as the growth proceeds, the phase change, everything is under microRNA regulation. So far as students of science, we have studied a lot about hormones, the phytohormones specifically. So the hormonal responses themselves are regulated by these microRNAs. And other than that, they are also known to regulate the transposons, and this helps in maintaining the chromosomal or the genomic organization, as well as the stress responses in plants. And what is, just to show you the power of these molecules, or what the impact of these regulations are, this is what an normal Arabidopsis plant looks like. You tweak one microRNA and then it can change into a leafy mass. This is the capacity of a microRNA. You change one and you can change the entire phenotype of the plant, from a Arabidopsis plant to a heavily vegetative structure. Or you tweak another one and then the plant can change into multiple shoots or bolts, and will completely turn into a heavily flowering structure. So this is the impact of the molecule. It is a very, very powerful molecule, and it holds the capacity to change the phenotype and the physiology of the plant. So just to make you understand how the microRNAs act, knowing that each molecule is powerful and there are many of them. So I picked up this classical slide which talks about the development of C. elegans. Now, in the insect, the life cycle can be divided into 4 broad stages: the L1, L2, the L3 or the L4 stage, and the adult stage. Now, if you look at the early larval stage, there is transcription of the Lin14 and the Lin28 transcripts, resulting in the formation of the respective proteins. And these respective proteins are required very early during the larval development. And as the larval stages progress, you see that there is the microRNA Lin4 will appear, and it will come and restrict the Lin14 and the Lin28 microRNA translation, so that the proteins are not formed. And this absence of the protein acts as a signal to push the insect from stage 1 to stage 2 or 3 or 4 of development. What you see in the lower panel is that during all 3 or the 4 stages, there is a transcript called Lin41, which is getting transcribed into a Lin41 protein. This protein acts as a repressor of another messenger RNA called Lin29. And this Lin29 message is constantly repressed in the larval stages. When the time comes to convert into an adult larval form, another microRNA Let7 will appear. This Let7 will also go and keep ensuring that the 14 and 28 are tightly repressed, so that the proteins are not formed, because these proteins are required only in the early stages of the life cycle. And at the same time, they will also go and block the Lin41 mRNA. So the Lin41 protein is no longer produced. Now, since this protein is not there, the suppression activity is removed, and we get the synthesis of Lin29 protein. And this is how sequential appearance of the microRNA will regulate the gene expression, so that the developmental timing of the insect can be tightly controlled. So this has been very quickly and very conveniently expressed or explained through this cartoon, but it took a lot of hard work and a lot of mutant analysis to reach this kind of model, so that it can be easily explained to you people. Now, coming back to plants, similarly, in plants, we have a battery of microRNAs which keep coming and disappearing